Hello, good afternoon, everyone. Hello, students, good afternoon. Welcome to Excel Connect. Have you joined? Can you hear my voice? Hello. Yes, good afternoon, everyone. Yes. You're able to hear my voice, right? Yes. Okay, fine. So, welcome everyone to Excel Connect. I'm Amrita. Today we'll be studying with Neural Control and Coordination chapter. Yes, good afternoon to everyone. So in previous class, we have taken 2 classes already. In the previous class, we studied with the neurons, like mainly with the human nervous system, where it was divided into 2 parts. That is central nervous system and the peripheral nervous system. Right? The central nervous system had 2 parts. Which were they? Brain and spinal cord. Right? Yes, good afternoon everyone. So what did we study in previous class? We studied about the different types in it. Central nervous system has 2 parts, that is brain and spinal cord. Whereas when it comes to the peripheral nervous system, it's on the lateral sides of the body. In that there are again autonomous nervous system and somatic nervous system. Right. Again, the autonomous nervous system was further divided into parasympathetic and sympathetic nervous system. When did the parasympathetic nervous system work? Can I get the answer? When the parasympathetic nervous system will work? In the normal condition. Whereas when it comes to the sympathetic nervous system, to react and expend energy in times of stress. So we saw that the sympathetic nervous system works in the stressful situations, right? There's one more classification of the somatic nervous system. Okay, that is, it's divided into sensory nervous system as well as motor nervous system. Yes, normal condition is parasympathetic division and sympathetic division during the stressful situation. Okay. So next one is with the somatic nervous system. The somatic nervous system works during carries messages from senses to the here. In that somatic nervous system we have sensory system. This sensory system works during carries messages from senses to the CNS. Okay, another one is there, that is the motor system. This motor system carries messages from CNS muscles to muscles and glands. Okay, so these things we studied in the previous class. Also we studied about the structure of neuron. Do you remember the structure? Fast we can brush up all the points. So what are the structures that we studied? What are these structures called as? What are these structures? College will start soon. Once you come here to collect the uniforms, at that time they would say the date. Okay, so these are the dendrites. Okay, and what is this? It's the nucleus. This is the cell body. And this large projection is called as? This large projection is the axon. We have this axon terminal. And then this is synaptic knob. Right, and the axon was covered by what? Axon was covered by Schwann cells. right? And the Schwann cell which had secreted some material, what was that? Remember? What was secreted from the Schwann cell? Myelin content, right? So the Schwann cell which had secreted myelin, which was circling this, which was covering the axon, was called as myelinated nerve fiber. Yeah, myelin, it formed the myelin sheath. If it has not secreted the myelin, then we call it as non-myelinated, right? Yes, myelinated nerve fibers are mainly found in the, where do we find if it is myelinated? There are two types, myelinated and the other one is non-myelinated. Okay, these are mainly found, these myelinated ones are mainly found in the central nervous system, that is the spinal cord and brain region. Okay, whereas when it comes to non-myelinated neurons, these are highly found in the, these, where do we find, any idea? It's found in somatic nervous system as well as the autonomous nervous system. This myelin sheath is just like how the electric wire will be there, how it is prevented from losing its signal, like the electrons, like from coming out, how it will be insulated. In the same way, the entire stretch of axon is covered by Schwann cells, which prevents the loss of signal. Okay, and here we studied what are these regions called as, where the Schwann cells are present and the gap between the Schwann cell. Previous class I explained you saying that we have the axon region, on that there will be Schwann cells which are laid over like this, and the gap that is created, those are these gaps. What do we call it as? Yes, these are called as node of Ranvier. And then we studied about the small granules that were found on this cell body, right? These small granules are called as Nissel's granules, right? These are called as Nissel's granules. And then followed by that, here these Nissel's granules are also found on the dendrites region also, but it is not found on the axon, right? What is meant by myelinated? Myelinated means we have the Schwann cell. Okay, Schwann cell gets squeezed and it forms an extended region, and from here it will start, from the plasma membrane of the Schwann cell, the extended region will start secreting a content called as myelin. Okay, and this myelin is transparent. Now what happens, these Schwann cells will cover the axon region. So what happens, this transparent Schwann cells will lie one above the other, completely covering the axon region. Okay, so that makes a myelinated neuron. Understood? Yes, cyton or the cell body, and then we spoke about Nissel's granules. So what are these Nissel's granules contain? It is made up of ribosomes and endoplasmic reticulum. Understood? So this is with the structure of neuron, which we have covered in the previous class. We'll move to the next topic. We were discussing about this in the last class. Till there we have done, we spoke about in depth, even we spoke about the visceral nervous system also in the previous class, where this, these are the neurons that are connecting the central nervous system to the visceral organs of our body. In today's class, we will be studying with the types of neuron. Yes, synaptic knob can also be called as a nerve ending. Yes, it's synaptic knob is at the terminal part of the neuron. It's at the nerve ending. Yes, beta. Okay, shall we move to the next topic, that is with the types of neuron? Yes, RER and ribosomes. Monisha, yes, correct. So now we will be moving to the next topic, that is with types of neuron. These are very important areas for your NEET exam. Like in the previous topic which I discussed, there the concept of this Nissel's granules, what is it present? 2 times it has come in NEET. In the same way, here this topic, types of neuron, is also very important with respect to the competitive exam. Okay. So in types of neuron, we have written the mainly 4 types. The first one is unipolar neuron. What difference can you see in comparison to the previous structure that we studied to this unipolar neuron? What difference can you see over here? What's the difference in this unipolar neuron? Is there any structural difference that you can see? Yes, you can see the difference. Yes, but what is the difference? Can you see the dendrites over here on the cell body? Is it present? No, right? So, unipolar neuron, it doesn't have any dendrites and it has one axon. How many axon is present? It has one axon and it has no dendrites. Yes, there are no dendrites. Okay, very good. So now, where do we find this type of unipolar neuron? These are found in the, for this, where it is found in the embryonic stage. Right? Yes, there are no dendrites, a cell body is present and we have a lengthy axon with axon terminal region. So what's the example? Where do we find this? These are found in the embryonic stage. Very good. Shall we move to the next one? Next one is with the bipolar neuron. Okay. So when it comes to the bipolar neuron, here it has its cell body and it has one axon, and this is from the cell body, it extended to form one dendrites. It extended to form one dendrite. It's not the axon region. This is the axon region and it extended here. A lengthy, it got extended and then it formed the dendrites region. So one dendrite it has. Okay. And see here, in the another end it should have axon terminal region, right? How we studied in the previous diagram, or over here. It should have axon terminal. But what it has sensory receptors. So you can find sensory receptors in the bipolar neuron. Got it? So it has polarity, like it has 2 regions it has. One is that, one region is with the dendrites and the other region is having the sensory receptors. Okay. This bipolar neuron, where do we find it? Yes, correct. Pratik, Manisha. Yes, right answers. These are found on the examples for this is example for this is retina of the eye. Okay. Yes. So unipolar neuron is found in the embryonic stage. Bipolar neuron is found in the retina of the eye. The speciality is that it has sensory receptors at one end. And next is pseudo-unipolar neuron. Okay. See, it is a cell body. It has one cell body. And this is the axon. It has axon. It has an axon region. Okay, and this axon is divided into, again, this one axon came and it got divided into 2 regions. Okay, so it has one axon and the same axon has divided. Okay, So this is with the pseudo unipolar neuron. Okay, and this pseudo unipolar, it has the axon terminal regions on both the end, right? It has axon terminal region. Can you see any dendrites over here? No, rather than that we have two region also, here as well as here. Both the side we have this terminal region, axon terminal region. So where do we find this? Yes, very good. These are found in the peripheral nervous system. Good. Yes, Pratik as well. Yeah, Pratik. Yeah, Pratiksha. Yes, the answers are correct. It's found in the PNS. Okay. Next, let's move to the next topic, that is with the multipolar neuron. This is that neuron which you have studied right from 8th standard and again in 10th and again in 11th standard you will be studying this, because this is found majority in our body. We are studying about this. Here it has many dendrites. How many dendrites it has? It has many dendrites. Okay, and it has one axon, and then it has axon terminal. So where do we find this? Mainly in cerebral cortex region. Okay, so where do we find this kind of neurons? We find in the cerebral cortex region, right? Okay, I hope now you have understood with the different types of neuron, that is the different structures, how the neurons are present: unipolar, bipolar, pseudo unipolar and multipolar neuron. I hope you have understood the topic now. Okay, now we shall move to the next topic. Okay, next we'll be studying about transmission of nerve impulse. Okay, so in previous diagrams you have studied the, here in this one you studied with the size structure of neuron, how it is, and here we had the axon terminal region, right? So now what happens if I say the impulse is coming from here? Okay, impulse got into the dendrites, the signal reached the dendrites, and from cell body it will pass
ಇದರ ಇತ್ತೆಂದು ಜನರೇಟ್ ಆಗ್ತಾ ಇದೆ ಕರೆಂಟ್ ಅನ್ನು ಜನರೇಟ್ ಆಗ್ತಾ
అట్లా పని చేసి పని దొరుకుతున్నాడు డైరెక్ట్
Okay, yes, we are back now. Fine. Yes, there were some glitches. We are back. Let's study. Let's continue with the topic, that is transmission of nerve impulse. Okay. Can you hear my voice? Everyone? You are able to hear my voice? Yes, don't be sad. We are back again. Yes, you are able to hear my voice, I guess. Yes, okay. So now we were studying with the transmission of nerve impulse. Okay, the transmission of nerve impulse takes place by electrochemical impulses. That means the transmission of impulses from one neuron to another and across one neuron itself will take place by the combination of electrical and chemical signals. Okay. Now, consider a neuron with its dendrites, axon region and its axon terminal region. Okay. So now when the impulses comes, it will be received by the dendrites. Okay. It will be received by the dendrites. It passes through the cell body across the axon. When it is passing across the axon, it is passed in the form of electrical signal. Here in the axon, it is passed in the form of electrical signal. When it reaches the axon terminal, this electrical signal gets converted and makes the vesicles over here. Okay, here you see the transmission from here, from the axon terminal region. When it passes to another neuron, here it passes by chemical signal. And this chemical signal will be received by the next dendrites. And these dendrites will take those signal. And again that signal will pass in the form of electrical signal and again back to chemical signal. Okay, this is how it passes. What exactly happens over here, how this electrical signal then got converted into chemical signal, and how this chemical signal next went to the next neuron as an electrical signal, will be studying now. Okay, so now what happened here, there was a gap between the two neurons. When the distance is bit more, then this chemical signal transfers. There are sometimes what happens, again here we have a neuron with an axon and axon terminal region, and again dendrites, axon terminal. In case if the distance between this axon terminal, that is the synaptic knob, and the dendrites of another cell is very less, here the distance is less, then in that place, instead of chemical signal, there passes electrical signal. Here it was electrical signal, then passed chemical signal, then again electrical signal. We'll be studying about those gaps also, like what do we call those gaps, everything I'll be explaining. Now just understand the transmission of nerve impulse across from one neuron to another neuron takes place by electrochemical impulses. Electrical signal, chemical signal, and again electrical signal. But here when it chemical signal is passing, then the distance between the two neurons should be more. Whereas when the distance between the axon terminal and the dendrite of another cell is very close, when they are in close proximity, electrical signal passes. Okay. In our body, most of the time chemical signal the transmission of impulses will be in the form of chemical signal, and this electrical signal is very rare. Okay, now I'll explain what exactly is happening over here, how this electrical signal is being transmitted into chemical signal, what happens in the axon terminal region, what is meant by synapse, everything we'll look into it now. This is the axon, okay, and this part is the axon terminal, fine, and here this part is the synaptic knob, right? This entire structure is the synaptic here knob, right? This entire structure is synaptic knob. Previously what the entire neuron we used to draw, now I am drawing only the terminal part. Yeah, axon ending, axon terminal region, synaptic knob. Now this region we can clearly say as this, as instead of saying like that we can say presynaptic membrane. This region is called as a presynaptic membrane, okay, and it can be another cell which is over here, or it can be the dendrite of another cell. It is called as postsynaptic membrane. Okay, we have presynaptic membrane and postsynaptic membrane, okay, and now here in this region we have a fluid filled space. It is called as synaptic cleft, okay, and how is this synaptic cleft? It's like a fluid filled space, let me write it that side so that it will be easy for me to explain, okay, and this region is called as the synaptic cleft, okay. Now here the combination of presynaptic membrane, synaptic cleft and postsynaptic membrane, all these three together we call it as a synapse. What do we call it as? Synapse. We have axon terminal, and in the synaptic knob region this part is called presynaptic membrane. It can be another cell or it can be another cell's dendrites. It is postsynaptic membrane, and there is a fluid filled space between, which is called as synaptic cleft. Totally the presynaptic membrane, synaptic cleft and the postsynaptic membrane together we call it as synapse. Got it? And now here there will be small vesicles. Okay, what are these? These are the vesicles. Okay, there are some small vesicles in the axon terminal region. Now what happens? An electrical impulse will come through the axon. Okay, what will happen? Electrical impulse will come through the axon, which we'll be studying in the next class, how electrical impulses is being transferred. Okay, consider now an electrical impulse is coming and it reached the axon terminal region. Now what happens? It causes something called as depolarization. When the electrical impulse comes to the axon terminal region, the charges or the ions over there gets changed. As a result, here in the axon terminal, there will be a channel, okay, which is called calcium gated channel. What do we have here? Calcium gated channels. Now what happens? Electrical impulse came to the axon, finally it reached the axon terminal region, and in the axon terminal region there was vesicles. Okay, and now what happened here? As soon as electrical impulse reached there, it caused the depolarization of the axon terminal region, because of which there was a channel called as calcium gated channel. Whenever you hear about channel, if there is a word gated along with that, then understand that will be closed. It need to open. There are different types of channels like mechanical gated channel, voltage gated channel. Okay, here we have written calcium gated channel or ligand gated channel. So what does that means? These channels will be in the closed form. Whenever there comes any signal, or then only it will get opened. Here it is calcium gated channel, which is a voltage gated channel. This belongs to voltage. What it means? When an electrical impulse comes, when it causes depolarize, which causes the, yeah, I'll be explaining. So this, there is a channels. Channels means what you have studied about in plasma membrane. You might have studied about extrinsic, intrinsic protein, etc. Right? So these are nothing but why they are there. It is there because they are acting like channels. Okay, so these channels here, this is voltage gated channel, which means whenever the voltage fluctuation comes, then the voltage gated channel which is closed will open. There are some mechanical gated channel. Whenever there is some pressure applied to that region, then the mechanical gated channel opens up. If I touch like this, I'm able to sense it, because when I touched it, pressure was there. That pressure made the mechanical gated channel to open. So there are different types. In the same way, here we have this calcium gated channel. When the electrical impulse came, calcium gated channel got open. Okay, now what happens? Through this, all the calcium ions will start to enter in. Okay, now what will this calcium ions which entered through this electrical impulse came? It made the calcium, it caused a depolarization, it caused the calcium gated channel to open. Now what will happen? This calcium gated channel made the calcium ions to enter inside. This will push all these vesicles towards the presynaptic membrane. Okay, the calcium ions will now make the vesicles to move towards the molecules called as neurotransmitters. These are filled with neurotransmitters. Even here also it is filled with neurotransmitters. Now from where did this neurotransmitter molecule was formed? Okay, we are having vesicles. From where it formed? This neurotransmitter molecules was formed in the cell body. Okay, we have cell, we have endoplasmic reticulum, we have ribosomes over there. So these neurotransmitter molecules was formed in the cell body, and it came through the axon, and these molecules are filled in these vesicles now. Okay, now with the vesicles which were in the axon terminal region, because of the calcium gated channel got opened, this calcium ions made the vesicles to come towards the presynaptic membrane. Now what happens? This neurotransmitter molecules will come and fuse with the presynaptic membrane. What will happen? Enagutelli? This will come and attaches, or it fuses with the presynaptic membrane. It fuses, and whatever the, okay. So now what happened? We have this vesicle which got fused with the membrane. Here one more vesicles. It will start moving. It will come here and it will fuse with the membrane. Which membrane? To which membrane it will come and fuse? Presynaptic or postsynaptic membrane? This will come and fuse with the presynaptic membrane. Okay, now when it comes and attaches to the presynaptic membrane, whatever the neurotransmitter molecules were there, they will be released to this, those molecules are released to the synaptic cleft. Okay, here fluid filled space is present, that's called synaptic cleft. Here all the neurotransmitters which was there inside the vesicles are now put into this synaptic cleft region. Okay, now what happens? This, it can be a dendrite or any other cell. It will be having small structures which are, here it has some receptor molecules. There will be some receptors. Okay. So they can ask you, where are the receptors found? You are able to see the receptors? Let me draw it like this. Okay. So there are receptors that are present on the postsynaptic membrane. Okay. Yeah, it is released to the synaptic cleft, and then there are receptors on this postsynaptic membrane where these neurotransmitter molecules will come and attach. Okay, these neurotransmitter molecules will come and attach to the receptors present over there. Yes, yes. Okay, so now we have this neurotransmitter molecules which got attached to the postsynaptic membrane. Okay, ma'am. Now what will happen if these neurotransmitter molecules, if it comes and attach? The main goal is transmission of the impulses. So electrical impulse came. Here it was neurotransmitter molecule. This is how the chemical signal, the message is passing through in the form of chemicals. That is in the form of this neurotransmitter. Okay. So these neurotransmitters will now attach to the receptor. What will happen next? Here there will be other channels. Okay, there will be other channels. They will get open. Okay, so let me write now whatever other molecules, it can be sodium, potassium, whatever ions will be there, they will start to enter through this, all these other channels which are present. Okay, here we saw electrical impulse came, and then the neurotransmitter, chemical impulse, it got converted into chemical impulse. These neurotransmitter molecules got released to the synaptic cleft. Finally, it got attached to the postsynaptic membrane, which caused the other ion channels that are present on the postsynaptic membrane to open up. It can lead to the influx of the any other ions like sodium, potassium, etc. Okay, this nerve cell is called as excitatory, right? This nerve cell is called as excitatory. Excitatory, or it's also called inhibitory. Excitatory, if it wants to send some signal from one place to another, then the nerve cell should have the role of excitatory. If I say it is acting like inhibitory, then it should be able to stop that neuron like functioning. It should slow down that neuron. Okay, ask the doubts, Baba, whatever you have. Okay, so excitatory example molecule is acetylcholine. What is this? This acetylcholine is one of the neurotransmitter. It's a neurotransmitter. And inhibitory, if it wants to slow down this passage of the signal. We have one more example for inhibitory, that is GABA. Okay, we have one more molecule which acts as an inhibitory. Yes, those are neurotransmitter molecules. Yes, correct. No, vesicles itself will not convert into the neurotransmitters. They both are different. Vesicles are like the vehicle in which these neurotransmitters are present. These are just there, these vesicles are there to transport the materials. Okay, yes, all are the examples for neurotransmitter. Acetylcholine is an example for excitatory action. If it needs to send the signal, then there is acetylcholine. If it wants to inhibit, we have GABA, gamma aminobutyric acid. Okay, this gives the inhibitory actions. Okay, now we have a picture over here. Okay, we have this axon terminal region. Okay, what are neurotransmitter? These are the chemical molecules. Okay, here previously I showed, no, these are the chemical molecules which are present so that the electrical signal need to be passed. These are the, yes, these are the chemical molecules that are present to send the signal. Here till here we got it in the electrical form. Now it has the chemicals in the form of neurotransmitters where these messages will pass from one place to another, from one axon terminal region to the next cell, that is to the next cell or to the dendrites. Okay, GABA is an inhibitory neurotransmitter. Shamant, yes, GABA is an inhibitory neurotransmitter. If if it doesn't want to send any information, or if that action need to be stopped, that means the neuron should slow down. That function is done by the GABA receptors. Delete. Yes. Okay. So now this is the same diagram with a bit more explanation. We have the axon terminal, that's axon, and this is the entire synaptic knob region, and we have, yes, already my student, yes, good. You are already in Excel. Yes, it's good that you are studying here as well as you are watching the online class. Fine. So here we have this axon terminal region where vesicles are present, vesicles move, and these vesicles are called as synaptic. Since it's in the synaptic region, we call it as synaptic vesicle. Okay. So now this synaptic vesicle comes, fuses with the presynaptic membrane, and here also calcium ions will come. Here is a calcium gated channel. Calcium will start to enter, okay, which will make these vesicles to come and fuse with the presynaptic. Neurotransmitters are released to the synaptic cleft, and they are attached to the receptors, which will make other ions to come inside this cell. Now, what will happen to the neurotransmitter that are released here if they want to, if the signal need to be stopped? Okay. Okay. How to stop that? If there are many neurotransmitters over here, then how can you make that neurotransmitters, like, it should stop its function? What will happen? Surrounding there is something called as glial cells. When we speak about the nervous system, it's not only just the neurons that are present over there. Okay, you can also see various cells such as astrocytes, oligodendrocytes, okay, glial cells. All these kind of cells totally makes up this, or that comprises the nervous system. So whatever the excess neurotransmitters will be there, whatever molecule, it can be the acetylcholine or it can be the GABA, after sending of the signal, still if there are this neurotransmitter molecule, then they should be removed from that synaptic vesicle, that synaptic cleft region. So now what will happen? When it is opening, you are asking about college, you need to come to the college, collect the uniform. At that time, they'll be informing you the date when the classes will start. If you are asking about the ion channel, that will open when the neurotransmitter molecules, when it attaches to the receptors on the postsynaptic membrane. So whatever the excessive neurotransmitters that are present, they will be retrieval of vesicular membrane from plasma membrane. So whatever happens, whatever these ions will be there, these will be taken up by the glial cells. What the glial cells will do, they will cause the enzymatic degradation. Okay, enzymes will be there in this. This is the picture of a glial cell. Okay, these glial cells will take up the excessive neurotransmitter inside, and there occurs enzymatic degradation. Okay. Date they will inform you soon, beta, once you need to come to the campus over here. College opening date, yeah, that's what, you need to come to the college here. At that time they will inform you. Now they are giving the uniforms, please collect them. Okay, so now these glial cells will take up all these neurotransmitters and they will degrade it, so that every signal there should be a stop when the signal need to be stopped. Understood, everyone? Yes. So this is how the neurotransmitters are transferred. So what all did we study in today's class? We studied, so these contents, the starting contents, nervous system, we studied in the first class, followed by that all the types, how it works, along with visceral nervous system we studied, and in the second class we studied with the structure of neuron, how it works, the different parts, what are the importance of Nissel's granules, what it does, everything we studied, and we studied what is meant by myelin, non-myelinated, and previous and today's class we studied with the types of neuron, very important with the perspective of NEET and other competitive exams, especially with how many dendrites it has, how many axon it has, whether if it is having dendrites, and in which location it is found. And followed by that, In today's class, we also studied about how the transmission of nerve impulses takes place. It's an electrochemical impulses. It transmits by electrical and chemical signal when the distance is far. When the distance is nearby, it passes in the form of electrical impulses, and these electrical impulses are very rare in our body, whereas the chemical signal and electrical signal combination is what happens most of the time in our body. Okay, and then followed by that we studied with the how the chemical impulses, this is with how the chemical signals get transferred from the synaptic knob to another neuron. Okay, only we studied with the chemical signal. We didn't study with how electrical signal will transfer. So in the next class we'll be studying this, how the electrical signal gets transferred across the axon. I hope you enjoyed today's class. Let's meet in the next class. Thank you.